system. And the peripheral nervous system we're going to talk about, right? A little bit. The classification. Central being central, it's going to be in the middle. It's just the brain and the cord. Okay. Peripheral nervous system is pretty much everything else. So we talked about the split of the two different ones. Basically, that little chart here. Okay. So you should know this. It's pretty difficult to kind of come up with a question on that, but kind of know the splitting there. If there's a question about, is it, parasympathetic, does it belong in the autonomic or somatic system? You should know that it belongs to the autonomic system, because the autonomic one is divided into two.
Can you repeat that?
So if I was to ask you, like, say, is the parasympathetic system part of the autonomic nervous system, or is it part of the somatic nervous system?
Okay.
You'll know that these are autonomic because you can't control them,
Okay.
whereas something like, muscle and stuff is going to be something you can control. Yes?
Do we need to know about the ENS? What?
We'll talk about the enteric nervous system in the gut, right? Are you referring to? Yeah. We'll talk about that during the GI lecture series. Basically, there's another kind of small nervous system sort of thing that's on its own, and that's in the GI system. But we won't talk about that. Okay, so functions of the nervous system, you pretty much know these. There's sensory, integrative, and motor functions. There's afferent and efferent nerves. Efferent are usually motor, right? Okay, so a little bit of histology of nerves. We talk about neurons, neuroglia, and the myelination. These are all pretty important. This is important. I would know all the different parts of an axon. If you were asked in the lab question, you would draw one, or if you were asked to point out something on the exam, you should know the different parts of it. Okay. But don't just know for your standard, axon. There's a bunch of different kinds we talked about, right? There's 3 different ones. Here there's the bipolar, the pseudo-unipolar, and the multipolar. So be able to kind of point out all those things on any one of those. So you can classify them based on their structure, right? So there's multipolar, and that means there's lots of dendrites. There's only one axon. Those are mostly seen in the brain and spinal cord. And there's bipolar. There's one dendrite and one axon. You don't see those everywhere. You see them in the ear, the eye. We talked about them in the eye, right? They're part of you get a photoreceptor, you have bipolar, and then you have the ganglion neurons that come out and go off your track, right? You definitely need to know that.
I have a question.
Yes.
Slide 4.
Yes.
Not that one. It shows the bipolar.
Oh, okay.
Or maybe it's the one right after.
This one?
Yeah. How can you? Well, if the picture is not that clear, what is it actually that you're looking at?
That's
It's
different.
so much different. Yeah.
Okay, so the location of the cell body in a bipolar neuron, you see the cell body is continuous with the axon here, and this is all a dendrite on this side, okay? Whereas over here, you kind of have a 2-part axon, where the body's kind of deviated off to the side. So you have a peripheral axon, and then you also have a central axon. Okay, that's going to go to the synaptic pole, the dendrite's up here. So over here, it's actually just one dendrite, okay, with all these branches on it, whereas here you have a bunch of different dendrites branching on. Whereas in multipolar, you have all these dendrites everywhere. Kind of all over the place, and then you have the cell body, which is surrounded by all the dendrites. You see the cell body over here. And then there's just one long axon. That's kind of the classic example of multipolar, like that other picture here. Multipolar. So there's a bunch of dendrites and they're all branched off the cell body there. Okay, and then you have the long axon with the synaptic terminal picture. Okay. And okay, so we talked about neuroglia as well. There's a bunch of different kinds. There's 4 types in the CNS, and 2 types in the SNS as well. And what do they do? So they participate in many activities involving neurons. Okay, structure, repair, myelination. I would know those. Okay, I would know those. And say there was a question about, which of these is not something that a neuroglia cell can do. Okay, so you definitely those 3 would be on there. And those would be all, things they can do, but there's something they can't do maybe. Okay. You don't need to know their names, that's fine. Astrocytes are kind of important. We talked about a few other times as well, so those are pretty much the only ones you would kind of be able to recognize. And then Schwann cells as well, we talked about in detail with respect to myelination. okay, and that's what we'll talk about next. And so you have the sheets formed, right? They're multi-layered. Pretty much those cells kind of the Schwann cells pretty much just wrap around the neuron and insulate it, and it increases the speed of electrical impulse conduction. And the spots between them are the nodes of Ranvier. Okay, and you get increased myelination with age from a baby to adulthood. Not like when you get really old, but like from baby to adulthood. Because that's what babies are always stumbling around and stuff. Their axons aren't particularly myelinated as much as in adults, and so their impulse conduction is not as quick, it's not as efficient, etc., etc. Okay, so note this diagram too. Okay, so you have clusters of neuron cell bodies or ganglia. We talk about several different types of ganglia throughout the body in lab and in lecture. And then we have a bundle of axons that pretty much makes up nerves. Okay, in the brain, most likely, and then also in other parts of the central nervous system like the cord, you have white matter and gray matter. So white matter is myelinated axons, and then gray matter is generally cell bodies, the dendrites, unmyelinated axons, axon terminals, and also nuclei, supporting cells. Okay, we talked quite a bit about action potentials as well. Does anybody want to talk about action potentials? Want to describe them? Someone on track? Okay, I'll do it then. It's my job. I probably should. I'm just trying to get out of doing my job, not having you guys do it, but it's okay. Okay, so electrical impulse messages are sent by way of action potentials. How does that happen? You have electrical charge being sent down an axon, basically. You have a charge switch of sodium, potassium ions, because they're positively charged, so the balance changes. When those balances change, the different amounts of them change, and the amounts change give a pulse that's sent by way of electrical conduction. Okay, so you have a resting membrane potential. Does anybody remember that resting membrane potential? No? Let's find it here. Visually you can see negative 90 where a cell rests at. Okay. I would know this. This is standard any neural class you have, you're going to know how an action potential works. And all the different phases of the action potential as well. So you'll have the opening of the sodium channels, okay? And then a bunch of sodium rushes into the cell. And since the cell's sitting at negative 90, and you're having all this positive charge come in, what happens if you're a negative charge and a bunch of positive comes to you? What's going to happen to you as a cell? You're going to become more negative or positive? More positive, right? Because more positive cells are coming into you. That's what's happening. Becoming way more positive, coming towards zero. Towards zero, okay? And eventually it goes away from zero, but in a positive direction. And then those sodium channels start to close, and the potassium channels start to open. And when that happens, potassium rushes out of the cell. Because remember, in the cell, there's a lot more potassium, and then there's more sodium on the outside. Okay? So you have this positive potassium leaving the cell, right? What's going to happen to you if you're hanging at a positive 35 and your positive potassium are leaving? You're going to go back towards zero. You're going to lose positive charge, meaning you're going to go towards zero and even lower. Okay? You're going to get more negative. Okay? And that happens to a point where it might even go a little farther down past negative 90. That's hyperpolarization. Eventually, those gates close as well. And then you have active transport that's going to swap out those potassium sodiums and restore balance at the negative 90 resting membrane potential. Okay, so know all the phases of that. Be able to identify, say there's a question with a graph like this and it points to this or something and asks, well, what's happening to the sodium channels here? What's happening to the potassium channels here? Okay. So again, there's leak channels, gated channels, okay? Leak channels are kind of always work, they're always on. And potassium's always kind of leaking out of the cell, right? And there's sodium's kind of always leaking in, but it's not much. Okay, and then you have gated channels, are the ones that we talked about pretty much in the action potential. They're voltage gated, okay? So the electrical change, the impulse, is what dictates if those guys are going to work. And that's seen here, again in this chart, where once you have a bit of a jump here, then those gates start to open, the sodium gates. And then once around up here, that's when all the potassium gates start to open because they're affected by this charge of around 35-ish, okay? So when the charge starts to change, that's when the shape of those channels start to change as well. And that's what, then they start to open, but then when they come back down here, they stop working. It's like, that's kind of where they're used to hanging out anyway, the potassium ones. So they'll start to change back, and that's why you have, that's why they close, okay? Nothing's coming over there telling them to close. It's dictated by this charge on the side.
What causes action potentials again?
What causes action potentials?
Yeah, like why does this happen?
Because of some sort of sensation or some sort of afferent impulse at the dendrites. Okay, they're the sensing bodies of the neuron. So when something happens there, some sort of change occurs, which is going to send a message down to the axon, and the axon is going to translate that by electrical charge down to the synaptic.
So it could be anything from pain to a cold.
Yeah, depending on the type of neuron, and depending on the type of, like we don't go into too much detail on the structure at the dendritic ends of a neuron, but they all work in different ways. They can work with pressure, all the different ways. They can have movement sensations or whatever it may be. Okay, so you have quicker impulse change by way of the nodes of NPA, the insulation. The charge can jump from space to space between those nodes, okay? It doesn't need to go slowly across the entire axon. You have the Schwann cells that are wrapped around, you can kind of hop. And if you can hop over, it's faster. Okay? So this just kind of shows the progressive picture of how an action potential ends up all the way over here, starting up here. Basically with some, what kind of stops on. Okay. Synaptic transmission, similar to what we did in muscle. Be able to kind of go through this, and you should know the different things that happen. Basically, when you have a charge come to the end here at a synaptic pole, you have calcium rushing in. Okay, because that charge that comes, calcium gates open. When calcium rushes in, it causes these vesicles to kind of come over to the end here, and they're going to release the neurotransmitters. And neurotransmitters are really important. We talked about a bunch of different kinds: acetylcholine, epinephrine. Just a bunch of different things. I mean, those are the two main ones we talked about. And we talked about them in sympathetic and autonomic nervous systems. We'll talk about them again when we get to that section. But just know there's a bunch of different kinds, and know how they're released by way of calcium at the synaptic pole. Okay? And then those neurotransmitters go to the postsynaptic cell.
Okay. The calcium enters the cell after the action potential is already finished.
Yeah, it comes down to there. It opens those voltages because they're voltage gated as well. So that charge comes down here. That charge, that positive charge, affects the shape of those calcium cells or those channels, and the channel will change a little bit, and then that's what will get the calcium coming. And all that calcium coming is positive charge, +2 charge. So eventually, that voltage is going to change again. That membrane potential is going to change there, and those calcium gates will shut because that change back to a different voltage is going to cause it to change conformation again. Does that make sense?
Yeah, is the action potential
Well, so what happens, you have that positive charge shooting all the way down, right? And then, so there's the sodium gates and the potassium gates. But remember, there's also active pumps that will restore those channels. So it's not like it travels back up and that charge is stored. So once it's, changes kind of here, better see me here. So as it's carrying this way, what's left back here, like on this side, so now all the sodium's inside and all the potassium essentially come out, right? Because all the sodium rushes in, potassium rushes out. But normally you have a lot of potassium inside and sodium outside. So there's those 2-3 pumps. Remember, they take out 2 sodium and 3 potassium, or sorry, 3 sodium, 2 potassium. They just change it back the other way. So those, because the cell wants to have more K inside and wants more Na outside. Okay. So since it goes the other way around during the action potential, those pumps will come and say, no, come on out sodium, and brings the potassium back inside the cell and restores it that way. But it doesn't happen, like it doesn't come all the way down and come all the way back. After this comes all the way down here, as soon as that starts going this way, those pumps over here are going to start working to bring back the membrane potential to -90. Because this part's now already past the action potential. The action potential is already going this way, right? And by the time it's over here, over here, these guys are already restoring themselves back to -90.
So when calcium is entering the cell, is it at -90 again?
So when that you can kind of take that and kind of think of that in its own part. That's at the pole. That doesn't go back up. It doesn't travel the other way. Because those gates have basically, they have a period where they can't shift for a while afterwards. We didn't really talk about it too much, but the potassium gates, they sit below -90 a little bit, and they kind of sit there for a minute, and they're really hard to reopen. Those gates and the sodium gates. Once they change conformation, they have a resting period, okay? That hyperpolarization period, and with that, it doesn't allow the charge to go back the other way. It keeps the charge going in one direction. So when this happens here, this is resting. But then when that charge shoots down here, that's when it changes right around here only, okay? And it's calcium. There's no sodium or potassium shoot here. Just those sodium and potassium gates, they stop around here. And then that charge changes. You still feel the effect of the charge right around the internal part of the cell there, the bulb, and that's what causes those calcium gates to open. Okay, so here's some of the neurotransmitters we talked about. You don't need to know them by name or anything, but I can refer to, I can ask, oh, a neurotransmitter, aspartate, but you'll know it's a neurotransmitter. It doesn't matter what it does or whatever, but if I'm going to ask you a question about neurotransmitters, I might use one of these as an example, or anything really for that matter. But the fact is, if it's going to be a question about a neurotransmitter, it's going to be something about this calcium, the synaptic vessels, coming to the postsynaptic cell. Okay, so know this concept more than Okay, so next is spinal cord and the brain, right? So this one was a good chunk of anatomy, even in the lecture, but nothing too big.
So you can do the anatomy part of that in the lab?
There'll be lab and lecture, probably. The lecture exam, since we went over some of the parts of the brain, more so in the lab, but I might throw up a picture of the brain and ask you to identify a portion of it. Some one portion that we talked about during the lecture, or some stuff we'll touch on even now. So, protective coverings of the spinal cord, know them. Know that there's 3 layers of connective tissue around the cord. Okay, there's the dura, arachnoid, and pia mater. Know the order they're in, identify them on pictures that are shown here, and be able to identify them on a model as well. We have a spinal cord model here somewhere, so know where they are. You identify them here. Pictures similar to these. Google a bunch of pictures. When I say ID them on a picture, Google a bunch of different pictures. Make sure you can identify them on ones that aren't here. You're not just familiar to on this particular one. Because there's other drawings that might be, instead of these colors, they could be green, red, and black or something. Or they could be a different shape or size. But you got to know the kind of general idea of the structure and be able to point them out. Okay. Don't just be familiar with this. Just like I said before, don't just be familiar with this text here. Write it out somewhere else. Think about it a little bit. Because you won't have these visual cues in front of you. You can study the crap out of these notes, but then when you have the exam, those notes aren't in front of you. It's kind of harder to recall this stuff. So talk it out with your shower wall, okay? Trust me, it works. Okay, spinal cord anatomy. So we talked about basically the fact that it's from the lower brain to the upper part of the second lumbar vertebrae. And that starts the claudia equina, where all of it starts to split out. And that's because the spinal cord is not as long as your spine, okay? So it starts to stretch during embryology, and towards the end it starts to string out. So that's at L2, okay? And that's important for stuff like lumbar punctures where you're taking CSF out of a patient or something. You want to go somewhere where there's stringy spinal cord, so you're not jabbing someone's spinal cord straight up and then paralyzing part of their body. Okay, so there's 2 grooves in the cord. There's the anterior median sulcus and the posterior median sulcus. Okay, be able to identify. And there's 31 pairs of spinal nerves. Okay, and there's 2 roots to the axons. Okay, you got a root here, you get the root here. Okay, and this is the ventral root, and this is the dorsal root in the back. Okay, so this would be like person with their this is the cord, and they're looking this way at you. Okay, dorsal behind. Okay, and then there's this one here, and they join. Okay, and they make what's actually the spinal nerve, and there's a spinal nerve on each side, right? Okay, mixed nerves. It's got sensory and motor once it comes out this way, but here they're divided.
So from a picture like that, the ventral is going to be the one closest to you?
Yeah. And you'll be able to tell because you'll have the anterior median fissure here.
Okay.
And remember, it's the H, the butterfly looking thing, with the central canal of it with cerebrospinal fluid in it, right? That's really important too. So yeah, you'll be able to tell basically also, you should be able to tell because remember those spinal nerves, they come out, they flare out, right, out of the thing. So they're flaring out this way. So they're flaring out anteriorly, right? Because they're going to go into your whole body. Like, let's see here. So the spine in the back, right? The spinal nerves are going to come out this way to different parts of your body, right? They fan out. But they're not going to fan out backwards because then what? You're going to, there's nothing sticking out your back. So they will flare out forward anteriorly, so you'll be able to tell just by that. And also you should be able to tell because there's that fissure there in the middle that kind of split, okay?
Right.
So just little things like that will help you figure it out. And also you'll see the dorsal horn is right. It's pretty much connected there to your dorsal root spinal nerve. Whereas over here you don't have the anterior or the lateral horn, or sorry, the ventral or the lateral horn aren't really connected to this section here. Just kind of little things like that to figure out. Central canal, CSF. Okay, and you would identify them on a histology slide as well, for the most part. For lab probably, you should know the lateral horn, the anterior horn, okay, and then also the, you don't need to know the lateral columns, but we're just going to talk about pretty much these guys here, and then the white matter is everything else. This is all the gray matter, the butterfly H.
So the lateral horn on the bottom is like towards the middle of the H?
Yes,
Yeah,
like
okay.
right there. Those ones are a bit more difficult. I'll make sure it's a picture. Like if I did a histo slide, it would look like this. It wouldn't be this kind of. I wouldn't ask you to tell me that's the lateral horn. That's pretty crappy. It flares out a little more. That's not the greatest. Somewhere else on the spinal cord, probably lower down. But it would be something like that. Okay. So we talked about these already. Okay, spinal nerves, 8, 12, 5, 5, 1. Okay, they're both sensory and motor. Diaphragms nerves. They exit the vertebral column via the intervertebral foramina. We were examining that on the spinal cord model there. All right, that could be a lab question. So a little bit about different plexuses. That's from a bunch of spinal nerves forming together. Okay. We didn't go into the crazy anatomy of those, but you will at some point go to brachial plexus when you go into more in-depth talk about the nerves in your next anatomy class. But know they exist, and they're a pain in the ass to learn. So if you want to familiarize yourself with these nerves now, the brachial plexus, go ahead. There's YouTube videos on how to memorize it and stuff, and how to draw it out. But for this class, you don't need to know that. Okay, we went over reflex, tapping into the knee, okay, how that works. It stays in the spinal cord. We talked about the inhibitory and excitatory neurons here. So know this pathway, be able to trace it back, know how it works, be able to describe it. Now the brain, there's the stem, diencephalon, cerebrum, and cerebellum. All different parts, all really important. Brain stem here. Okay, you get midbrain, pons, medulla. Know them, know how to be able to identify them. Be able to tell me even what order they come in. That's important. You should know. That's kind of anatomy based, but it's very less important. The cerebellum in the back, the nasty looking thing we always talk about, right? For posture and balance. And you have the diencephalon and cerebral hemispheres. There's two of them in most people.
What do you mean by order?
Like in what order they come in. Like if
Just like in structure?
yeah, in structure. You should be able to identify, not necessarily, they're not gonna have like defining borders on them, but like if I was to put a picture of one and ask for the three labels and tell me what order they came in, you should be able to identify it. Not necessarily exactly where they are, but you should know what order they come in, because they all have different significances. And especially talking about primitive function and things, mostly down in the medulla. Okay, the blood-brain barrier is pretty important. Protects the brain from harmful stuff, for the most part. Some things can pass through there. Oxygen, CO2, alcohol can, which is not doing its job, it's not protecting against alcohol. But for most stuff, it'll stop from going through. And it's consisting of tightly sealed blood capillaries and astrocytes. Okay, so that's where astrocytes come back, those are neuropia. Those are the ones we talked about, and the Schwann cells, okay? Those are the two types. I would expect you to know. I mean, if I asked the function of one of them, if I asked about, what kind of insulated cell neuron, it's not going to be these astrocytes, it's the Schwann cells, okay? If I ask which ones are part of the blood-brain barrier, you're going to know it's not Schwann cells, it's astrocytes. Okay, CSF, you should know its functions. It's clear, colorless, that's fine. Circulation of the CSF is important because if you have a block in the circulation of CSF, you get what's called hydrocephalus. And those ventricles, remember the ventricle model there? Those can fill up with CSF, high pressure, and we look at CT scan too, where it starts to expand, and pushes your cerebrum outward. The brain gets pushed up against your skull, and it makes your brain shrunken, but it's just all that pressure inside of here. So it goes to 4 different ventricles, okay, and also travels through the central canal, all right, and it's produced by the choroid plexus at the walls of the ventricles, that's important, and it's also reabsorbed by the arachnoid villi. So if something goes wrong with one of these, if you can't reabsorb it, or you're producing too much, you can end up with a lot of CSF, again, pushing, high pressure inside in the canal, and in the ventricles, being something like hydrocephalus. Look at the ventricles here, okay, so you should know the anatomy of the ventricles pretty well. You have the lateral ventricles, right? Then you have the third ventricle, and then there's the cerebral aqueduct, which is important. That goes to the fourth ventricle. Okay, so if there's some sort of occlusion here, and these guys are swelling up like crazy, this one, the fourth will be fine. The central canal will be fine. But these are going to swell up quite a bit. Okay, so say you were given a picture of some really swollen ventricles, which I would tell you that they were swollen ventricles. I wouldn't expect you to be able to do that on your own. And these were really swollen. This one wasn't. Like, where's the lesion at? It's going to be the cerebral aqueduct, right? Or if this guy was really swollen, where's the lesion at? Probably somewhere below, but it could be above as well. It'll be really clear. Trust me, if I ask you a question like that. But know the anatomy of this, okay? Know that the cerebral aqueduct goes to the fourth ventricle, connects the third to the fourth. Okay. Okay, so then 3 parts of brainstem, broke them down a little bit. Medulla's down here, pons, midbrain, that's a nice, pretty colored picture. Okay, so let's know a little bit about the medulla. The cranial nerves that come off it, it's 8 through 12, or part of 8 at least. Okay, and the numbering goes from high up with the olfactory 1 down to 12. Okay, so obviously since midbrain, the medulla is the last, or the first part of the brainstem, but it's farthest away from the top of the brain. So cranial nerves, the top is where they start, so it goes down to 12. So it's going to have 12 to start with, and all the way up to the middle of 8. You can see that on the picture here. Okay. So you have the medulla here. Okay. Cranial nerve 1 starts up here with the olfactory bulb, so 1 through 12 all the way down. Since medulla is the lowest of brainstem, it'll probably have 12 in it, right? It absolutely does. That's 12 through 8.
Lecture questionnaire or not?
Could be either one. But analysis is not fun. Good to go. Okay, next is pons. Okay. Breathing control components, that's important for that, okay? And there's a lot of nuclear for voluntary movement, right? Not too important for this class. Don't worry about it too much. Midbrain is more important because it has what? Has what? No. Midbrain has, it's not part of the thalamus. So midbrain has the cerebral peduncles. What are those? What are those? They connect the brainstem to the cerebellum, right? That's important. Then you have the diencephalon, which has 3 parts: the thalamus, pineal gland, and then also the basal hypothalamus, which controls everything. And we talked about this in endocrine, so don't worry about it here, but worry about it in endocrine. And species you just know as well, a little bit of what they do, just briefly.
So the cerebral peduncles connect the brainstem to the cerebellum.
Remember how the cerebellum kind of hangs off the back of the brainstem and hugs it, and right underneath the back of your big brain, your cerebrum? So here, see how it hangs off? It's nasty looking thing? And your big brain's up here, your brainstem here. Well, it's connected to the brainstem. It's hanging on there by the cerebral conductors. Cerebellum, or cerebrum, sorry. Cerebellum. This is a picture of the cerebrum, but the cerebellum down here. So this is what we're talking about here. Know what it's for, okay? Know how it works, know how it gets input from everyone, where you are in space at any given time, pressures, sensations, everything. So it can kind of cross-check with your brain about what it's actually doing, and it allows for smooth movement as well. Now the cerebrum. So there's gyri, fissure, sulci. The ones we mainly talk about are the splits here. We talked about the central sulcus here in the middle. And on one side you have the motor cortex and the somatosensory cortex on the other side. Okay. We talked about how the occipital lobe is primarily visual. Okay. Then you have taste and speech and things kind of in the parietal area. The frontal is mostly association and involved with emotions. And then auditory, as you can tell, the temporals on the side. You've got auditory there next to the ear, and also some smell as well. Okay? So mostly hearing, mostly visual, a little bit of other stuff, a little bit of other stuff here. These are really important, and they're separated by the central sulcus.
Separated by what?
Central sulcus? It's not labeled here. It's labeled on the. So if I was to give you a picture or something like this, I would expect you to be able to tell me where these two are for sure. Which section is visual? You should know that. Which section is hearing is pretty important. These guys, not as much. But the speech area, we talked about Broca's and Wernicke's that come up at the end of this lecture. You can see that as part of the thing. Okay, limbic system, the emotional brain. Okay, it's important. Functional area, sensory, which is pretty much what we talked about here. So know this. Again, visual area, hearing area. Good, tell me. There's the central sulcus here. Okay. There's the premotor cortex again, and then the somatosensory cortex on this side. And we broke it down by the homunculus here, right? This one's the kind of bigger picture. So notice that in the sensory section, parts of your body that are pretty sensitive have dedicated more real estate along the sulcus here, okay? Or along this piece of the brain, basically, like the lips, and then other parts like your hands. Same with motor. Things that are really fine motor have a lot more real estate as well. We would identify kind of, not necessarily the order of things on here, but if something, if I was to ask you about, let's say your toes or something, they don't have much real estate. You should be able to tell me that, they'll have less space. Whereas something like your lips would have a lot of space. Okay, Broca's area we talked about, and then Wernicke's as well. You don't have this. I think these 2 or 3 slides might not have been on the handout part, but don't worry about them. The only thing you need to really know is about Broca's and Wernicke's. Did you have this? The cranial nerves?
Mine stopped at this.
Did you guys have this? Yeah. I think, I had re-uploaded it, so maybe you printed it out before that. You weren't here for this one either, right?
No.
Okay. Yeah, I re-uploaded it, but it has this on it, and you should know the cranial nerves. Talk about those in piece of detail right there. This will tell you everything you need to know about the cranial nerves. Know their numbers, know the order, and I told you to look up like cool mnemonics to figure out how to remember their order. Okay. There's a bunch of them online. That was it for that.
And the test will be like all of chapter 9 questions and all of chapter 10.
Most likely, yeah. I figure that's kind of helps you guys out, keep things in order. If I scattered them out, it's kind of tough because we jump a lot of concepts. Like going from a blood question to a brain question is kind of a pain in the read. Most physiology classes go this way too. You guys want to take like 5 minutes? Yes, so it's been 45 minutes. Take 5 minutes, 10 minutes, and we'll come back.